Hello Shishas, welcome to today's lecture on cell biology. Let us start with the introductory part. I want you to recall the things you studied in your previous class, that is 8th class. I hope you remember this fact: the cell was discovered by Robert Hooke, that he found in the specific sample of plant which is considered as the oak tree. Here he tried to isolate the stem, the cork of it, the outer layer, and he put under his own microscope, and it has revealed him the empty spaces which are called as the cell. In Latin this is called as cellulae, and which was translated in English as the cells. And then you can see the first living thing, whatever Robert Hooke observed, it is not considered as a living cell, but still he named as an empty cell, but it has a lot of things inside which cause the living things. And then we have the other scientist called Antony von Leeuwenhoek, so who was able to discover the first living cell. And Antony von Leeuwenhoek called those cells that he observed, like the sperm cells, the Orbices cells, all of them he observed under the tiniest microscope he prepared, which is of nearly 10x resolution, and he named them as animalcules. So later those were called as the microorganisms, and then you know that we have a big field of understanding the microbes called as microbiology. So all this credit goes to Antony von Leeuwenhoek. And next, when there was such discoveries happening one by one, and biology has demanded to come up with a theory for the cells alone. So this theory got a lot of observation, and then it required a lot of scientific field. So all of it became very mandatory. So at the time you can see two gentlemen, Sleeden and Schwann. So these two people came into the picture, and they tried to give the theory, the important theory called cell theory, to prove that biology is also possible to develop such theories. And Sleeden and Schwann, both of them are zoologists and botanists in their individual fields, and they have come up with few points which are very close to both living forms, that is plants and animals. So first they gave the cell is a functional and structural unit of all living organisms, and they even tell this point without studying it. So produced by pre-existing cells, and all activities of cells are controlled by the nucleus. And next is organisms are made up of maybe one cell or many other cells. And in class many times I used to illustrate this: whenever you say something is a cell, it should supposed to consist of three different particles or three different things. So let me illustrate you again here. So when you say something is a cell, so first it is supposed to have the cell membrane, and it should supposed to have the cytoplasm, and even it should supposed to have what we call as the nucleus. So if these three things are missing, so something which is even classified or considered as a living thing, it won't be perfectly called as living thing. So this has become an exception in case of viruses. So viruses exceptionally, they are not considered as living things, as they only have some genetic material. It doesn't compromise any three components of cells. And then there is one more confusion arised when they said all the activities of cells are controlled by nucleus. We know that nucleus have DNA. That's nothing but the genetic material that's being transferred from the species to species from the parents. And how far some of the cell organelles can contain its own genetic materials. And even these cell organelles are called as semi-autonomous organelles. So these two questions became most difficult things to be answered, but later on years, so though all of them are answered. So now we're going to look at what actually deals with study of cell. So we have a specific field called cell biology that deals with the structural and functional aspects of cell, and we have cytology that deals with structure alone, And then to understand a cell in more elaborated way, you should know something about the microscopy. So microscopy is a study or the field that studies about the fine structure by using the microscopes. Sorry, it's just a mistake. The microscope is a tool that is used to study the fine structure. We know that our eyes cannot penetrate through the microscopic cells in all the living things. Hence, we should supposed to use the lenses that helps us to observe the tiniest things, and we'll know more about them. And now you can see the two important terms you should know while you do microscopy are the magnification and then the resolving power. When you say magnification is one of the important aspects of a microscopy or microscope, where you have to look at it can be considered as a ratio between the retinal image and the actual image. And the resolving power is a unique characteristics that is used to enhance the study of objects. And whenever you say the resolving power, you can consider something like, when you say the resolving power, you have two close objects. So let's say you're observing the cell membrane in case of the plant, and even you can see there is an adjacent cell wall which is strong enough, which is really thick enough. When you have the resolving power, then only you can be able to understand these two close objects. Alright, so that's what called as the resolving power. When you try to differentiate between two close objects, it is called as the resolving power. The resolving power is one of the best enhancements, I can say, for the understanding of cell and its many of structures. Let's get back to the points that we are doing with. And these are very common microscopes you can look at in our schools. The first is called as the light microscopes, where the source of light is exactly the sunlight or the artificial electric bulb also. And then if you look at the ability of this light microscope maximum, it may give you the resolution about 300 to 1500 times, and then the resolving power is 0.25 micrometers. That means you can distinguish the objects which are nearly 0.25 micrometers. So beyond that, this microscope can't help you. And one more important thing is the light microscopes are very useful in observing the living things. Right? Whenever you want to observe any living thing, so you can use the light microscope. Alright, we have another type of microscope called as the electron microscope. The light microscope has a lot of disadvantages. In order to come over them, the electron microscopes are used. So the electron microscopes have a lot of applications, and we can even look at the microscope in very well manner. Do you know who discovered the microscope for the first time? I mean, the light microscope obviously discovered for the first time. So the two scientists who involved in making the microscope is far earlier than discovering the cell. So the scientist who discovered the cell is the microscope, the compound microscope, is Zacharias Janssen. H. Janssen and Z. Janssen, so both of them are involved in developing the first microscopes. But still, believe me, it's after 1590, it took nearly 60 years for Robert Hooke to observe the first cell. Okay, next we're going to look at the electron microscope. So electron microscope is discovered by two scientists again, so Knoll and Ruska. Maybe quite funny names, can't remember them. So Knoll and Ruska. So these two scientists discovered the electron microscopes for first time, I think, in early 1950s, and these things have changed the dimension of understanding the cells. As electron microscopes use the electrons as the illuminating source, so they gave more and more clarity of the objects that we are observing. And you can see the magnification has raised from thousands to lakhs, again based on what type of images the electron microscopes produced. We have classified them into two major types. So one is called as transmission electron microscope and scanning electron microscope. The scanning electron microscope will be useful to see the three-dimensional images, and the transmission electron microscope will be useful to produce two-dimensional images. And again, the scientist has to choose what is the requirement for the study of the cell. If you want to observe the smaller particles like proteins, he should supposed to observe the structures, because structures are exactly going to provide the information how they're going to work. Okay, and then when you look at the transmission electron microscope, here you need only how the objects are arranged. Okay, so that's why the TEM and on SEM are like the various purposes, and it will be chosen by the scientist. And if you look at the transmission electron microscope, has more magnification, and when someone goes for a scanning electron microscope, the resolution will be high and the magnification is less. Right, these about electron microscope, and all of this is quite important part for you to understand how these are going to be used to study the cells. So light microscope, the properties, the magnification power and resolving power. Electron microscope, two types, TEM, SEM. And now we're going to look at the units of microscopy. The microscopy units are very essential things when you consider them. Whenever you observe something under electron microscope or any microscope, you should supposed to provide the information at which resolution exactly you have observed it. I'll show you some of these samples. So whenever you try to observe any sample of microscope, the objects has to be mentioned with certain units. If you look at certain units, so meter is very common thing which is visible, and then the millimeter, which is found in the scale. So most of the objects which are in millimeter range, we can see them with our naked eyes. And then after that particular limit, our eyes can't observe these micrometers. So micrometer is nearly 10 power minus 6 meters, and then nanometers, and the short form is nm, it's nearly 10 power minus 9 meters, and then picometers, pm, it's about 10 power minus 12 meters, and angstrom is about 10 power minus 10. And picometer is like very limited used unit, I can say, but mostly you can see the angstrom and nanometers are used. And this is a picture of the compound microscope. You can see it has a lot of lenses arranged at different angles, so which gives the better visibility of the sample that you put on the slide, we can say. And now we're going to look at some of the okay, I think you can do this in your class. I'm going to show you some of the images, so the principles how these microscopes are going to work. The microscope, the first type of electron microscope, you can see the lens arranged, the eyepiece, the first one, the objective, and then the specimen is kept in between, and then optical condenser. So the light will be condensed by this, and then focusing knob. The focusing knob is very much useful to focus exactly the sample region. Alright, and then you can see illumination. Sometimes even this could be the source of sunlight also. There will be small reflecting glass, and this where you can see the sperm cells, which is not much clear. When you use this transmission electron microscope, you can see the electron beams are being produced. Then the condenser is here, and specimen is kept at a different place, and then the objective lens here, and then projector lens here, and the scientist can be able to view from this region. And then you can see the scanning electron microscope, so the one which produces three-dimensional images. So here you can see electron gun which starts producing the electron beams, and then the condenser is going to filter them, and the scanning electron magnets are here, and then the detector is going to catch the pictures or images are produced, and it's going to pass to the fluorescent screen, and here they can be able to observe it. You can see how different, the same thing. So here the sperm cells are observed, and here the transmission electron microscope showing the sperm cell, and finally you can see the scanning electron microscope showing the image, the same image at different dimension. So that's it from me today. From this lecture we'll try to continue with the next topic soon.